Well, thank you, Clemens, for this very kind introduction. And I just want to say briefly that I'm, for multiple reasons, actually very, very happy to be here. So first, I think I've been here at this conference maybe in the iRNA track 8 times, 9 times. I forget exactly the number, but very many times. And it's been very formative, I think, and I've made very many connections that were very fruitful for my career here. So I'm very thankful about that. Also, I used to study about 50 kilometers that way in Grenoble or wherever southeast is. I'm just going to say that is southeast. But so it's also in that sense sort of a homecoming here. So I will be talking about splicing, and this is a cover that we actually, when I say we, it's actually Luisa who made it in 2012 for the ENCODE papers. And basically, what he meant to say that here is that a lot of the splicing reactions are already occurring while the polymerase is still transcribing the end of the gene. So you see still 2 introns remain, but here this intron is gone, this one is gone, this is gone, this is gone, and this is gone. So splicing happens mostly co-transcription. Today I'll talk more about the cell type diversity of isoform expression. And so just to point out that this is Manhattan, this is where the university is located, this is where postdoc housing is located on this little tiny island called Roosevelt Island, actually just next to Manhattan, but very calm. So it's almost like a village next to Manhattan. Anyway, what we do is we use these long-read technologies to investigate the brain. We devise new assays, new applications for diseases, and then the algorithms and software that is needed to interpret this new kind of data, because very often when you have a new assay, you don't really have these off-the-shelf software pieces that would actually solve all your problems. So that's what we do. One more piece of introduction. So we can think of splicing as the process that takes pre-messenger RNA, that takes the gibberish, actually, that is pre-messenger RNA, locates the exons, removes the intron in between so that we get a final message. It was night in October. We can all understand what that means. Now, when the cell actually removed this last intron here, it may also realize, oops, there is a second exon here. Remove the intron surrounding it, and now we get a second message for tonight in the lonesome October. And so these two messages are actually quite similar. It's still about October, still about night being dark and this kind of thing. But there's a slight change in connotation, okay? Because the first phrase is just any regular phrase. The second phrase comes out of a poem by Edgar Allan Poe, okay? So that means this is one way to think about alternative splicing. Sort of, let's say, the slight modulation, more efficient, maybe more binding of the protein to something, but still overall the same message. But there's other ways to think about alternative splicing. So consider this phrase here: Monday I have facts. With just a little bit of alternative splicing, I can turn that into Monday I have alternative facts. A phrase that was made famous by Kellyanne Conway in the United States, and by that she actually meant pretty much the opposite. Okay? She was asked, can we at least agree on some facts? And she said, no, I'll give you alternative facts. And the interesting point is actually that happens in biology. So this is the Fas receptor here, a very famous gene. There's one transmembrane domain which is encoded by an alternative exon. And when you express this isoform with this transmembrane domain here, you will have a cell surface receptor that when the apoptosis signal comes, will accept that signal, and the cell will happily commit cellular suicide. And when that other isoform is expressed, this protein will, quote unquote, float around in the cell. And now, when the apoptosis signal comes, the cell will simply ignore it. So on a cellular level, it's really a difference between life and death. So I don't have the slide here, but there's actually data out of Harvard saying that there's actually quite a few isoform pairs from genes that behave that way. Not the majority, but something like 20% that really have these dramatic differences, and then another 30% that have quite strong differences, another 30 less, and then another maybe 10, 20 that have nothing. And so when we started this lab, we basically thought long and hard about how to untangle basically the complexity of this beast here and also of the mouse brain. And the starting point was basically to say that, whenever you have these signals, for example, DNA methylation or gene expression, RNA isoform usage, protein expression, fundamentally in a complex tissue like the brain, it is actually a linear combination stemming from many different cell types: neurons, ependymal cells, oligodendrocytes, astrocytes, microglia, blood vessel cells. and potentially all their subtypes, because many of these have many subtypes, and potentially even single cells, if you think about diversity of single cells, which is a kind of a different topic, but can also play. And so, we thought long and hard how to deal with this, and in the beginning we played a lot around with cell sorting and long-read sequencing. In the end, we came up with this method that we call scissor-seq, so single cell isoform RNA sequencing. Later on, actually, PacBio came up with a similar one. They dropped the R, so I think they pronounced it scissor-seq. But it's basically there are strong similarities between these methods. And I should point out that this project was led by Ishan Gupta and Paul Collier, but many people contributed to it. So I would invite you to read the author list, because there were many contributions that were crucial that are not mentioned here. And so, to make a long story short, we basically sit on top of single cell sequencing that I assume many people know already here, and then take basically unfragmented barcoded cDNA, do long-read sequencing, and here we actually did PacBio and Nanopore, but this was done and published in 2018, so done in 2016-17. So the Nanopore data at that point didn't look that great, so we really focused on the PacBio here. And so we locate barcodes in these reads, and according to the barcodes, we can say that these reads come from individual cells, which, according to short reads, have already been assigned to a cluster of cells here, and they're named microglia according to the expression of this TMAM119. And so we can say that this is a microglia read. We can do the same for the second one, which turns out to be a neuronal read. And this was done here at P1 cerebellum. Now we wrote 2 follow-up papers on that, one a software package in 2021, and I'll talk more about that in detail, and then the software that actually generates these plots that we've been using for a long time. Every line here is one molecule. Okay, they're grouped together because they come from one cluster. There's 3 blue clusters here. They're all neurons. Blue for us is always neurons. And you see that you have these orange alternative exons that are actually quite specific of the neurons here. This one, this one, this one, this one, this one to a certain extent, with differing levels for the different neuron types, and then this one again. When you go to microglia, you don't see any of these inclusion of these exons, and so therefore you really have this added cell type specificity on isoform level that is not there on gene expression level. Because on gene expression level, they're all expressed, otherwise we wouldn't be able to plot them anyway. And so you can download this, and so these, in conjunction, these two software packages let you produce exactly that. And so I'm going to talk now a little bit more about the statistics of this, and this was led by Anushka Joglekar, but again, many people contributed to it, and there's a software package called scissor-seq. And so the fundamental thought about this was now that we did long-read sequencing, which in many cases gives us full-length isoforms. Not always, but in many. And so, but until then, the statistics that we had been using were all on single exons, which fundamentally is, has been motivated by short-read sequencing. And, of course, if you do short-read sequencing, that's exactly what you should do. And there's many great packages, including from many people in the audience. But for long-read sequencing, it felt like we were sort of besides the point by focusing on single exons. Now, what Anushka basically argued was to say, well, if we have basically this hypothetical gene with all these long reads, and I'm going to skip over a lot of details here, we should not reason in terms of exon in, exon out per condition, but we should really talk about all the different isoforms. So what we have now is this 2x(n) table where we have n isoforms for a gene, and then the 2 conditions that we're interested in, which can be 2 cell types or the same cell type in case control studies, or the same cell types in 2 brain regions, and so on. The interesting point is that now you have one test per gene that takes into account pretty much anything that can differentiate isoforms. So TSS has taken into account every single alternative acceptor, every single alternative donor, every alternative exon, every poly-A site, well, the one poly-A site. We didn't put in RNA editing, but we could. And once you do this for direct RNA sequencing, you could, in theory, also put other RNA modifications. And what you showed is that in a comparison of 2 brain regions, this was PFC and hippocampus, so planning and memory in very early development, so 7 days after birth for the mouse. If she went by these exon tests, she had only 30 genes where something happened, and these isoform tests actually gave us 400. So part of it, I would say a smaller part, is maybe a little bit of statistical advantages because of the correction for multiple testing, but most of it is actually because now we have all these different biological types of alternative isoform usage that are covered by the same test. And so in the same paper, we also devised this other method we call slide isoform sequencing, or slice-o-seq, where basically the barcodes are not used in order to differentiate individual cells, but spatial regions. This was done in the mouse brain. So this is a slice here of the mouse brain. Up here you have the cortical regions. This here is the hippocampus, and that's the choroid plexus. And I'll forget about the other regions because I'm going to focus on that for the rest of the talk. And so it's sort of like doing 3,000 separated RNA long-read sequencing experiments by different regions in the brain. And so I just want to point out two kinds of models of changes of alternative splicing that we saw in the brain. And so here in these plots, now for these plots, we're just going to focus on single exons. Yellow means a PSI value of 100%, black means 0%, so complete skipping of the exon. And here in clathrin A, you can see this exon, which is 100% included in the hippocampus, and then it's basically completely skipped in the choroid plexus. The interesting point here is that the switch happens exactly as you progress from the hippocampus into the choroid plexus. So it's a switch in splicing that is determined by leaving one brain structure and going into the next. Okay, and here's another example, PKM, again, a very similar picture, and these are mutually exclusive exons, so the other exons do the opposite. However, there's precise exceptions. So for example, here in the hippocampus, you see mostly black, mostly skipping, but then there's just a little bit of yellow inclusion at precise pockets here. We're still investigating those to understand how that actually happened. But another model of alternative splicing that we saw was basically this gradual shift. So here you see a lot of inclusion in the cortex, and you see less and less inclusion as you go through the hippocampus, and slowly but surely you end up in this area where there's really a lot of skipping. So now it's not the frontier from one brain region to the next that matters, but it's this gradual shift. Okay, so it's a different paradigm of brain region-specific alternative splicing. Now, everything we did until here was basically in the mouse brain. And the mouse brain, in a sense, is beautiful because you can sacrifice a mouse at 12 o'clock and at 12:05 you can have fresh cells and they're on your machine and everything is good. Human brain doesn't work like that for very obvious reasons. You don't, or you rarely ever get fresh human brain. And
want to tell us something?
I won't go much more into it, but what happens to human brains is actually that usually there's a large postmortem interval, and then they're frozen down, and usually they disappear for quite some years in the brain bank, and then you get them. Now that means this regular approach of isolating complete cells is now impossible. Okay. Now, the workaround that many people have worked on and that has done a huge favor to the field is single nuclei sequencing. So you basically now extract single nuclei, which for gene expression is wonderful because you get a lot of reads coming from the intron, from the exon. It doesn't matter actually. It still means that the gene was producing some RNA. For us, it's really a nuisance when we're talking about exon combinations, full-length isoforms. So we came up with this technology to actually overcome these things. And this is a project that was led by Simon Hardwick, Wen Hu, and Anushka Jogalkar. Again, many people contributed to it. And we call it single nuclei isoform RNA sequencing. So I'll talk a little bit about the technology. And so what's actually happening when you do single nuclei long-read sequencing is you will produce three kinds of molecules. The first one is the one we really love. You get all your exons here, and you get the barcodes, the UMI and the barcode here, and some sequencing and PCR adapters, and that's really beautiful because you know which cell they're coming from, you have the exon combinations, all is good. Unfortunately, there's a lot of other molecules that are being produced. So the first one is kind of obvious, and I think a lot of people are aware of that, that basically the poly-DT primer can pick up A-rich regions within introns. And when the intron is large, you just get then 2 kb or something like that of intronic sequences represented in cDNA. And if that's not what you want, then that's a problem. The third molecule is something that actually fewer people are aware of, because when you do Illumina library prep, it's heavily biased against. But if you do long-read sequencing, now you're getting them. And that's when the TSO that's made to make the second strand is actually initiating first strand synthesis. And then you get this beautiful molecule, potentially with all the exons, but you have TSOs at both ends and no barcodes. So you don't know which kind of cell they came from. And so basically we devised methods to get rid of that, and I'm going to go quite quickly over that. The first one is basically a linear amplification from this black element, which only exists when there are barcodes. So we don't really get rid of the third one, but we don't amplify it. And then the second step is we use exon probes to enrich for the molecules that actually have any resemblance to known exons. And thereby we now strongly enrich for reads that have at least one known exon, and it turns out that those are often the ones that have multiple exons. And then we do long-read sequencing, either PacBio or ONT. They're now both usable with different, PacBio still has a higher correctness or lower error rate. ONT gives you more reads, at least on the PromethION, and both improving on where it's difficult. ONT on the sequencing error, PacBio on the throughput, different methods, but in principle they're both usable. And we did this now in a human cortex, and you find all the cell types you would imagine: excitatory neurons, inhibitory neurons, microglia, astrocytes, oligodendrocytes, and so on. And we could basically show that in terms of barcode detection, over the naive single nuclease sequencing, we had a 2.2-fold increase. And in terms of, so this is the first bar compared to the last bar, and in terms of having actually some exonic sequence, we had a 3.3-fold increase. Together, that should be giving you 7.2-fold. It actually gave us 7.5-fold, so there's a slight dependency between these two processes that we actually don't fully understand, but it's very slight. And that means that an experiment that previously would have cost us $30,000 now costs $4,000. Right? And now we can start thinking about doing multiple samples and doing interesting biology. Now, in this paper, we basically looked at a couple of interesting genes. And so what you see here is that the reads, it's still single nuclei, they're not completely full length. Right? So there's something missing here. Like all these reads don't make it to the very first exon. But there's quite a lot of reads that have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 exons. So that's already beautiful when you want to talk about exon combinations, about isoforms, about how polyacid works together with an exon, which is something we did. We didn't really look into. I'm going to skip over today. But here I just want to point out that now we can define what we consider variable exons. So this exon here is included in 100% of molecules from astrocytes. A little bit also in oligodendrocytes and OPCs, so other types of glia. Then you go to the neurons, and it's completely skipped. Okay, so the delta here would be, I don't know, the delta would be something like 95% minus 10% would be maybe 85. So that's a dramatically cell type specific exon. And this one, I think it's this one, might have been one of the other ones, was actually interesting because it was an exon whose inclusion is associated to autism spectrum disorder. Okay, so, and that means, I forget now if it's up or down. If it's up, well, it would logically come from excitatory or inhibitory neurons, maybe from oligodendrocytes, obviously not from astrocytes. If it's down, it would have come from one of these cell types here. And so we looked a little bit at that and looked at exons that were associated to different diseases. And here we looked at the ones from autism spectrum disorder, and the ones that are associated are heavily cell type specific compared to the ones that are not associated. So autism spectrum disorder associated exons are really very cell type specific, and there's some research pointing in that direction. Now, in ALS, we also found a significant difference, not as strong as for ASD, but it was significant. For schizophrenia, we did not find it. Now, I want to point out one caveat, that these exon sets came from different publications. People did different experiments, different statistics to get them. So I'm not sure if it's 100% comparable, but I think what stands to reason is that ASD is heavily cell type specific. ALS is cell type specific. I'm not sure if it's actually less than ASD. That might be because of the origin of the exons. But there is something, and in schizophrenia, we just did not find it. Now, on the right here are now the autism spectrum associated exons. And here we have 4 cell types: excitatory neurons, inhibitory neurons, astrocytes, oligodendrocytes. And you see a lot of exons highly included here. Red means highly included in all 4 cell types. So if these are down in autism, it's a very interesting question in which cell type were they down. They could have been in any single cell type that could have been down, or in all 4 cell types, or in any subset of cell types. In other ones, for example, these ones here, if they're down in autism, that could have happened only in astrocytes and only in oligodendrocytes. And now you have all these patterns, they're low everywhere, they may be just high in one cell type, but not in the others. So depending on what the pattern of inclusion in all these different cell types is, you can ask now very different questions about what's happening in disease. Okay, so long story short, the pattern of cell type specific inclusion in healthy individuals is important. And so the last part I want to talk about is work that we did for the NIH Brain Initiative, and was led by Anushka Joglika, Wen Hu, and Bei Zhang. Again, many people contributed to it. It's all unpublished, so please don't scoop us. That would be much appreciated. So fundamentally, what we did here is to try to get a feeling, a census of cell type specific isoform expression across the brain. And the brain is, of course, many things, many brain regions, and we did also for 2 brain regions a bunch of developmental time points. P21 is when mice open their eyes, very important for the visual cortex, of course. And so now we basically can ask for the time-dependent developmental regulation, the space, so different brain regions, and the cell types and subtypes. And so this was mostly done with nanopore. There's also quite a bit of PacBio. In the beginning, we were really doubting very much that we could assign complete isoforms to individual reads. Now, this work by Andrea and Alla actually made us really think that now we can do it. It's, they published or made this software that we call isoform, and it's really very, very specific, which means there's not a lot of false positives, which is one thing that we saw early on in the field. And I'm happy to talk more about this and how we actually learned what are the error sources. But not for now. So what we can do now is we can have basically here for excitatory neurons in the hippocampus at 4 different time points. We have all these inclusion patterns, and you see that this exon starts off to be included in maybe 20% of cases, then 50, then gradually they disappear, and then they come back in adulthood, P56. And if you look at oligodendrocytes, it's basically always gone. And what that means here is that these exons that we thought were really typically neuronal, and the exclusion would be typical glial, is actually not true. Okay. Now at P28, the skipping of the exon occurs equally in glia and in neurons. Okay. So that means when you say something like glial or neuron-specific exons or inclusion, you should really heavily think about the time point that you're talking about. Okay. The time point really matters. And so for each cell type now, we can look at these different sources of variation. So an isoform can vary by region, meaning the same cell type in 2 regions. Here we're focusing on excitatory neurons. It can focus by subtype, and it can focus by age. And so if you're by subtype, dot is an isoform, you will end up in this triangle. By age, you will end up in this triangle. By region, you will end up in this triangle. And so when what we saw for excitatory neurons here is that there was a lot in the subtypes and a bit by region, less by age. Now, we're especially interested in genes where we have 2 isoforms that are in different triangles. The middle triangle means you have equal variation, which means mostly low variation across all 3 dimensions. And so we asked, how often do we actually find genes that have 2 isoforms in 2 different quadrants? And we see most often that we have these 2 isoforms in the central triangle and in this one, a little bit in the center triangle and the upper one, which is region variation, and a little bit of also in region and subtype. Okay, now we can do this for multiple cell types. And what we see is, for example, by region, there's quite a lot of variation in oligodendrocytes and also in immune cells, and less so in astrocytes. And by subtype, again, oligodendrocytes are quite high. I wouldn't interpret this because we just didn't have enough resolution to distinguish subtypes of immune cells. But again, a little bit less, although there's some in astrocytes. Now, the interesting statement is actually that when you put this all together, for about 3/4 of the genes, you can find some pair of isoforms that in one pair of conditions has very strong regulation. Okay, so that means there's dramatic isoform regulation going on. It doesn't mean that if you have just two conditions that there will be necessarily 3/4 of the genes having a different, but if you consider all these 20, 30 conditions, there will be one where this gene is changing something, poly-A sites, TSS, return of exons, and so on. This doesn't even take into account the difference between oligodendrocytes and astrocytes of neurons, so it's probably a lower bound. Now, I'm running out of time, so I'm going to summarize these plots really quickly. Just to say, when you start with a mouse data, and define what we call extremely variable exons, so that means there's two conditions between which they vary with a delta psi of 0.75. So 90% inclusion in one cell type, 15 in another, or 90% in one cell type in the hippocampus and the same cell type in the cortical plexus of Mendeleev. If you now do all these experiments again in human hippocampi, and you put on the x-axis how much cell type variability there is, what we find is that usually what we observe in mouse is also present in human. Okay, so you can largely transfer your observations from mouse to human. Not entirely, but largely. If you go the other way around, you can find a lot of cell type specific exons in human that behave the same way in the mouse, but there's an added number of cell type specific human exons that are cell type specific, a bit less so, and those you cannot see in the mouse. Okay, so in summary, what you can model in the mouse is transferred well to human, but there's added things in human that you cannot model so well in the mouse. With that, I'd like to thank you for listening. Yes, all the former lab members, mentors, and collaborators at Weill Cornell, especially here, the 4 blue ones that really helped me a lot. Collaborators outside of Weill Cornell, Luisa for the plots and the funding sources. And I'll leave you with this slide. I'm not going to go through it. It's sort of the summary. You're welcome to take a picture. And if you have any questions, I'll try to answer them as well as I can. Questions?
Thank you, Hagen. That was a very nice talk. So you've shown us that there's great splicing diversity based on age, cell subtype, region, but there's also a lot of splicing events in neurons that are dependent on cellular activity. So have you thought about looking into this and how to do that?
So we have thought about it. Now, in this data set, it's not as easy, but it's not impossible. So there are some marker genes that are expressed with cellular activity. So you could eventually, segregate cells that have a lot of this marker gene expression from the ones that don't. Of course, it's not a perfect, easily a perfect cutoff where you say like above this there is cellular activity, below this. So that would be one approximation from this, let's say, from this tissue-based approach. But of course, you could also force cells, to be active, and this would be more the kind of approach that you will do in a test tube, and then simply ask what changes, and then you would not necessarily need the single-cell approach. We've thought about it. We have not done it. It's actually, there's a very interesting question about this, also about how long these cells then are actually active. Right? Because if it's a short impulse, the question is, it's not straightforward to me how long it would take till that would be till that would lead to changes in alternative splicing. Is it even the same for every gene? There could be some genes that are reacting immediately in alternative splicing. There could be other genes which take a while. So it's I think there's an entire field that one could explore, and where many PhDs actually could ask different questions. So I don't have the answer, but I think it's studyable.
Hi, Hagen. Maybe I missed it, but have you tried to compare the cell type specific splicing patterns between mouse and human?
Yeah. So basically, the statement was that here in mouse we had this for example, this class of exons here, I went very, very fast over that, so this column, in this column red means is that there's cell type specificity in adulthood, okay? The other column means that there's cell type specificity early on in development. So these guys here are actually the ones that have cell type specificity early on and then maintain it all the time. When we trace them into human and look at their cell type specificity, we find this big peak at 1. So their cell type specificity is really transferred all the way to human. Okay. Then we have other sets here that have cell type specificity early on in development and that lose it in adulthood. Now, when we look in adult hippocampi, they don't have it either. That's the yellow line here. Right? So again, that means the absence of cell type specificity in adult hippocampi is maintained. Whether the cell type specificity early on in development is maintained in human, we don't know because we didn't do early time points. You know, this P14, P21, we would need some adolescent human brains. We didn't have them. Not impossible, but we didn't have them. And then other ones here are the ones that actually gain it, and they are equally maintained here in adult hippocampi. So, but the second statement was when you go back, there's about 40% of cell type specific exons in human. So these ones here, I'm not going to go into the details, but this picture means perfect cell type specificity in human. You look in mouse, perfect ones. This picture here means 1.45-fold more that are somewhat cell type specific in human, and they're not doing that in mouse. Okay, so that means there's two subsets, and this leads me to the statement, you can more or less transfer mouse to human, but there's added things happening in human that you cannot transfer to mouse.